Alright, so you went through transcription. Transcription was making a segment of mRNA from the DNA gene. And now we're going to go through the second part of protein synthesis, which is translation. So, translation is the second step of protein synthesis. And remember, protein synthesis is using a DNA sequence to make a protein. And two steps are transcription and translation. So, translation, these notes are not going to be nearly as long because we already talked about the different types of RNA, and now we're going to talk about rRNA and tRNA. Last one we only focused on mRNA. So where this happens is this happens in the cytoplasm. At a ribosome. Why it happens is because we need to make proteins. Alright, and then what happens is the ribosomes read the mRNA code in sets of 3 and tells tRNA to bring amino acids to the ribosome. The ribosome then puts the amino acids together to make a protein. Alright, so you don't have a picture here, but I do because I put a picture here. So first, before we get to the picture, let's just show you some of these here. I can do that one. We'll go for this one. Alright, so here's our mRNA. It's single-stranded, it has U's, so we know it's mRNA, not DNA, but this was copied from the DNA in the nucleus. And then we have...
Translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown.
Alright, the only thing you need to know about this is this ribosome here, right here, attaches to the mRNA. And then the tRNA also attaches to the mRNA. You see how the tRNA has 3 bases on it that attach to the mRNA? And then the tRNA brings us this thing right here, which is amino acid.
The 70S ribosome has 2 sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond.
So the tRNA, the next tRNA comes with a different amino acid, or it could be the same depending on the sequence right here, 3 bases in the mRNA. And then the ribosome combines those 2 amino acids together.
The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
So the ribosome leaves, and now we have a protein here. A whole bunch of amino acids put together makes up a protein. And that's basically what it is. So if I show it to you on a picture here, crap. So we're going to use this picture here. So there's some non-English words here, but we'll get the picture here. So this right here, this single strand, this really long single strand, this is the mRNA. Alright, and the mRNA, where did it come from? It came from the nucleus. So I'm just going to draw a circle out here that shows that the mRNA came from the nucleus. Alright, so here's our nucleus. Oops. Alright, so there's our nucleus. The mRNA came from the nucleus and then comes here to the ribosome. I think that even though that's spelled differently, I think you guys can still figure that this is a ribosome. The ribosome is reading the bases 3 at a time. Alright, so ribosome reads 3 bases at a time. That's called a codon, but you're not going to really need to know that. You just need to know that it reads 3 at a time. Those 3 bases signal a tRNA. So these are the tRNA. It's already labeled for you. And the tRNA brings the amino acids to the ribosome. So this little circle up here is an amino acid. Those amino acids then get stuck together to form a protein. So really, you're just going to have to be able to recognize this stuff and be able to explain it. Alright, so I'm going to write these in steps then too. So I want you to draw a picture and use words to describe the steps of translation. So 1, mRNA binds to the ribosome. 2, the ribosome reads the mRNA 3 bases at a time. 3, the tRNA brings the correct amino acid, and it's based on those 3 bases, to the ribosome. And then 4, the amino acids are put together to make a protein. Alright, easy. So now, where do these codons come into play? So we're going to talk about codons here, and this is on your second page. So codons are those set of 3 bases that code for amino acids. Alright, so here's a codon chart. So as you can see, there's several different amino acids, and they're all coded by 3 bases. 1, 2, 3. 1, 2, 3. So what you're going to get is, if you get a set of 3 bases, or 4 bases, a set of 3 bases, you're going to have to use this chart to determine what amino acid it has. Now it's pretty easy. This one's really easy because it has a set of 3 bases. All you have to do is find it. So if I give you the mRNA, and I give you UUC, UGA, AGG, CGG. So the first thing to make it easier for you is either draw lines or put spaces in the sets of 3 so you can read them easier. Alright, now we just go up to the chart and find UUC. So UUC is, you see how the bracket is around it, so it stands for PHE. So the first amino acid that you're going to bring is PHE. UGA is stop. So we would stop, but I'm not going to actually stop. I'm going to keep going. But that's a stop codon. That means we stop the amino acid sequence. AGG is ARG. And then CGG is ARG again. So you see that some of them are coded by different sets of 3. And that would be our protein. Now I know it stops right in the middle of it, but we'll just ignore that. Alright, so that's how you use that. Now the tricky part is if they give you DNA, and remember you know it's DNA that if it has bases, T's as the bases. So I'm already dividing these up in sets of 3. You can't go right from the DNA to here because there's no T's. These are not the DNA bases, these are the mRNA bases. So the first thing you have to do, remember, just like we did in the protein synthesis step, is we have to transfer this into mRNA with translation, transcription. So UAG, AAC, AAG, CAG. Alright, now we can go up here and go to UAG. UAG is stop again. So I'm not very good at this picking non-stops things. AAC, it's ASN. AAG, it's that, and CAG is this. Alright, so again, what you have to do is just look at the 3 bases and find what there are. But if you get DNA, you have to make sure you go to mRNA and then to the amino acids. And this same difference, if you're given amino acids, so if I'm given Val, THR, and ARG, SER. Then I have to go, if they're asked for the DNA sequence, then you have to get the mRNA first. THR, ACC, and you can just pick one of the 3. And then you have to go to the DNA, CEA, TGG, TCG. Alright, so that's the kind of trick that you have to make sure you have to recognize it as DNA or mRNA, and then you have to make sure you go with the intermediate step before you go right to the amino acid. A lot of people forget that.